And so then after olfaction, we can talk about audition. This will be the transduction of sound waves into a neural signal. We have three different areas of the ear. We have the external ear, which has the auricle, your auditory or ear canal, and your tympanic membrane, also known as the eardrum. And then we have the middle ear, which consists of your malleus, incus, and stapes, your three inner ear ossicles, or middle ear ossicles, if you will. And then we have our inner ear, and that consists of our cochlea and our vestibule. And our vestibule has the semicircular canals attached to it, and these will be associated with equilibrium and balance, which we'll get to shortly. And then your cochlea is associated with transmission of sound, or transduction of sound, I should say. So zooming in a little bit closer on the inner ear, your equilibrium or balance originates from the vestibule. So that's this organ here. And this vestibule and the semicircular canals make synapse with a vestibular nerve. And your audition is associated with your cochlea. And once the sound makes its way through the cochlea, it sends a signal to the cochlear nerve. And the cochlear nerve and vestibular nerve congress. They don't actually merge together, but they do end up forming one bundle of nerves that go on to the brain. And this is called the vestibular cochlear nerve. And so our auditory pathway that we've kind of started on at this point, the sound waves would be entering through the auricle. It would guide them in and into the auditory canal. The auditory canal would direct those sound waves to the tympanic membrane, which would vibrate that tympanic membrane. And the tympanic membrane would then make contact with the three middle ear ossicles. And those three middle ear ossicles amplify those sound waves such that they connect to the cochlea via the oval window. And then that displaces fluid that is inside that cochlea, and it moves that fluid like sound waves or ocean waves. The mechanical energy of moving the bones and the sound waves moving those bones is converted into electrical energy with organs inside the cochlea. So the fluid moves through the cochlea and it displaces hair cells, which are connected to neurons, and those hair cells open up ion channels that elicit an action potential in those neurons that they're connected to. And so that nerve that they're connected to is the cochlear nerve, and this will then transmit signals to the brain that can get processed and interpreted for meaning, so disseminating what sounds we're hearing. And so zooming in a little bit further, we have our organ of hearing. This is called our organ of Corti. Okay, so sound waves are going to pass through, vibrate on the tympanic membrane, amplify by the ossicles, and then make contact with this oval window of the cochlea. The sound waves travel through into this scala vestibuli, and so basically think of, I'll have another view here shortly, but think of this as one long continuous tube, and once it gets into the scala vestibuli, it starts to displace this vestibular membrane, which has a different fluid inside of it, and that displaces the tinctorial membrane here, which then displaces the organ of Corti. And that is how we get an action potential to be produced, which makes contact with the nerve and then goes on to the brain for association or processing. So the scala vestibuli connects directly to the scala tympani continuously, and throughout this continuous tube, sound waves move through the liquid and displace the membrane, causing bending of the hair cells in the organ of Corti and producing an action potential. And so what we're seeing here is movement of sound waves through the fluid that move the tinctorial membrane in hair cells. So you have the sound waves coming through, they vibrate and move the middle ear ossicles, and you can see the sound waves start to come in. They displace the membrane here, and that displacement displaces this membrane. And then that membrane displaces hair cells. Those hair cells notice the movement, and that movement depolarizes them. It changes the potential of these cells, which send an action potential to the cochlear nerve. And then that tells the brain, oh, I'm hearing something. So that mechanical movement of the membrane stimulated an electrical impulse, an electrical energy, in the cochlear nerve. So mechanical energy was converted into electrical energy to tell us we were hearing something. Perception of sound. All right, so it travels through this scala vestibuli all the way until it's in the scala tympani, so that it just kind of meets right here. It's just this continuous tube, and then it makes its way back out through the round window. All right, so this, I'll have it on the next one here. Basically, what you're seeing here is transmission of the electrical signal from the cochlear nerve going on to the primary auditory cortex in the temporal lobe, which we don't exactly see right here, but that's what's exactly going to happen.
Welcome to 2 Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the cochlea. When sound waves travel through the canal of our ear, they hit the tympanic membrane or eardrum and cause it to vibrate. This vibration prompts movement in the ossicles, a trio of tiny bones that transmit the vibration to a structure called the oval window, which sits in the wall of the cochlea. The cochlea is a tiny coiled structure in the inner ear that resembles a snail shell. The interior of the cochlea consists of 3 fluid-filled canals that run parallel to one another: the scala vestibuli, the scala media, and the scala tympani. The scala vestibuli and scala tympani contain a fluid called perilymph, and the scala media contains a fluid called endolymph. When the oval window is depressed by the ossicles, it creates waves that travel through the fluid of the cochlea. These waves cause a structure called the basilar membrane to move as well. To visualize the function of the basilar membrane, it can be helpful to imagine the cochlea uncoiled. When waves flow through the fluid in the cochlea, they create small waves within the basilar membrane itself that travel down the membrane. Different sections of the basilar membrane respond to different frequencies of sound, and as the waves progress down the membrane, they reach their peak at the part of the membrane that responds to the frequency of the sound wave created by the original stimulus. In this way, the basilar membrane accurately translates the frequency of sounds picked up by the ear into representative neural activity that can be sent to the brain. The translation of the movement of the basilar membrane into electrical impulses occurs in the organ of corti, which is the receptor organ of the ear. It sits atop the basilar membrane and contains receptor cells known as hair cells. Hair cells are so named because protruding from the top of each cell is a collection of small hairs called stereocilia. When the basilar membrane vibrates, this causes movement of the hair cells and their stereocilia. Movement of the stereocilia opens ion channels and causes the release of neurotransmitters to propagate the auditory signal to the vestibulocochlear nerve, which will carry the information regarding the auditory stimulus to the brain to be analyzed and perceived.
So that fed directly into equilibrium now. And so if you want, you can go back and watch that video again and or look into it even further just so that you can have your own explanation of the stepwise process of how we get to audition or hearing. So with equilibrium, we're talking about balance, and this originates in receptors that we can find in the vestibule and also in the semicircular canals. So we have 2 different types of equilibrium that we can look at, static and dynamic. When it comes to static equilibrium, we're looking at head position. So whether the head is moving or not by acceleration or just tilting. So it's sensed by the vestibule. There are 2 different organs in there. You don't need to know them, but it's basically this utricle and this saccule, and they have macula tissue in them. They're responding to gravity and acceleration. So gravity forces this top heavy otolith to bend the nerve or the hair cells. So think of it as if you're standing upright, the otoliths are pushing pressure down, and so that's making a synapse onto these nerves, telling your body that we're upright. Well, then you push your head down. Well, gravity forces these otoliths down, forcing the hair down, forcing them to send a signal to your brain saying, my head is tilted down. And so this is the same thing as when you're moving in a car. If you're moving really fast, these would be pushed back the other way, and then your body is telling you, it'll make synapse on here telling your brain that, hey, we're moving, like we're not stationary. But basically, bending of these hair cells is what actually stimulates the electrical signals for the interpretation of position by the central nervous system. And so again, this goes to that vestibulocochlear nerve. Then we have dynamic equilibrium. This is looking at your head movement. And so if you're spinning around, your semicircular canals are going to sense this. You have 3 different ones responding to 3 different planes, such as anterior, lateral, and posterior. They all have an ampullae in them. And looking closer at what this ampullae contains is there's a paralymph in here, just like in your your cochlea. And so this is actually called endolymph. This endolymph gets displaced whenever we move, and so that's what this is circled here is behind is moving due to inertia or movement. And so this, you moving your head this way, moves the liquid that way, which moves the hair cells this way, along with the liquid, which tells the nerve or fires an action potential in these nerve cells or hair cells to stimulate an electrical signal to go on to the central nervous system for interpretation of position. All right, so the actual movement of the head moves the endolymph, and the moving of the endolymph moves the cupula, which contains the hair cells, and the movement of those hair cells depolarize and then send an electrical signal or action potential to the central nervous system via the vestibulocochlear nerve. So I have another little video that we can check out here.
Welcome to 2 Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the vestibular system. The vestibular system is a sensory system responsible for providing our brain with information about motion, head position, and spatial orientation. It is also involved with motor functions that allow us to keep our balance, stabilize our head and body during movement, and maintain posture. The main components of the vestibular system are found in the inner ear in a system of compartments called the vestibular labyrinth, which is continuous with the cochlea. The vestibular labyrinth contains the semicircular canals, which are 3 tubes that are each situated in a plane in which the head can rotate. Each of the canals can detect one of the following head movements: nodding up and down, shaking side to side, or tilting left and right. The semicircular canals are filled with a fluid called endolymph. When the head is rotated, it causes the movement of endolymph through the canal that corresponds to the plane of movement. The endolymph flows into an expansion of the canal called the ampulla, within which there are hair cells, the sensory receptors of the vestibular system. At the top of each hair cell is a collection of small hairs called stereocilia. The movement of the endolymph causes movement of these stereocilia, which leads to the release of neurotransmitters to send information about the plane of movement to the brain. The vestibular system uses 2 other organs, known as the otolith organs, to detect forward and backward movements and gravitational forces. There are 2 otolith organs in the vestibular labyrinth: the utricle, which detects movements in the horizontal plane, and the saccule, which detects movements in the vertical plane. Within the utricle and saccule, hair cells detect movement when crystals of calcium carbonate called otoconia shift in response to it, leading to movement in the layers below the otoconia and displacement of hair cells.